Welcome to the Deep Dive, where we take a stack of complex sources and extract the most essential, fascinating nuggets of knowledge you need to be well-informed.
Today we're tackling a system that's rarely the star of the show.
The urinary system, and specifically the kidney. And, if you look at the sheer physics of what it accomplishes every day, it's just astonishing.
It really is. We're talking about true industrial-level chemical processing.
Absolutely. So consider this fact that really hooks you. You only have about 3 liters of plasma in your circulation, yet your kidneys filter that entire volume approximately 60 times a day.
60 times.
They churn out a staggering 180 liters of raw filtrate in just 24 hours. I mean, that is intense.
It's the very definition of metabolic housekeeping. The urinary, or renal system, is composed of the paired kidneys, the plumbing, so the ureters and urethra, and then the storage tank, the bladder.
But the kidneys are doing the heavy lifting.
Oh, yeah. They perform not 1 or 2, but 5 major functions to keep you in equilibrium.
Okay, let's unpack those 5 roles, because I think most people only think about waste disposal.
Well, waste disposal is number 1 for sure. The removal of nitrogenous metabolic waste like urea, creatinine, and uric acid, but they are also the supreme regulators.
Regulators of what, specifically?
Role 2, maintaining your critical fluid and electrolyte balance. So managing sodium, potassium, calcium, all essential for nerve and muscle function.
And the third role?
They handle the long-term regulation of your blood pH, your acid-base balance. They can either conserve or eliminate excess acid, like hydrogen ions, or base, like bicarbonate ions.
So waste, salt and water, and acid-base balance. That's 3.
Aye.
What about the other 2? They don't seem as directly linked to, making urine.
Correct. Role 4. They are absolutely central to maintaining your systemic blood pressure.
How so?
By managing blood volume and by secreting some powerful enzymes and hormones that control the constriction of your blood vessels.
And the last one?
Role 5. The regulation of red blood cell production. They release a hormone, erythropoietin, which tells your bone marrow to make more red blood cells. They are truly the silent multitaskers of the torso.
So our mission today is to take a deep dive into the engineering genius of the nephron, that microscopic filtering unit, and explore the surprising evolutionary history that explains its remarkable efficiency, especially as we examine the common challenges it faces. So let's start with the physical structure. The kidneys are tucked away against the back wall of the abdomen, the left one a little higher than the right, and inside, the functional unit is the nephron.
Yes, and the process of making urine is a 3-step journey through that nephron. First, filtration, then reabsorption, which is taking back all the good stuff, and finally, secretion, actively dumping unwanted waste into the tube.
And it all begins with glomerular filtration.
Right, which is achieved by this incredible structure called the filtration membrane.
How does the kidney manage to separate the tiny things it wants to discard from the big vital things it needs to hold on to? It must come down to that membrane.
It's an exquisite 3-layered, highly selective barrier. The first layer is the inside wall of the capillary, the fenestrated endothelial cells.
Fenestrated, meaning they have pores.
Exactly, little windows. They're large enough to let water and small solutes through, but they are still small enough to prevent blood cells and platelets from ever leaving the circulation.
Okay, so the first screen keeps the red stuff in the blood. What's next?
The next layer is the basal lamina. It's a layer of gel and collagen fibers. This meshwork physically prevents most of the large plasma proteins from passing through.
But here's the critical part I found interesting. The fibers in this lamina carry a negative electrical charge.
Yes, that's the key.
Why is that so important? I mean, the size exclusion already seems to work.
It's an insurance policy, a really clever one. Most of your key plasma proteins, especially albumin, are also negatively charged. So that negative charge on the filter acts like this massive electrostatic shield, actively repelling the proteins.
Like 2 magnets pushing each other away.
Precisely. It's functional redundancy. Nature is making absolutely sure you don't lose the proteins that keep water where it belongs in your blood.
And the final, finest filter.
That's the visceral layer of the capsule, made of cells called podocytes. They have these intricate foot-like extensions called pedicels that wrap around the capillaries.
And they interweave.
They interweave to form these tiny filtration slits. These slits are the final, tightest barrier, making sure only the smallest molecules, water, electrolytes, glucose, amino acids, and our nitrogenous wastes get through.
So that massive output, 180 liters a day, is quantified by the glomerular filtration rate, or GFR, averages about 125 milliliters per minute.
And that efficient rate is achieved with a surprisingly low net filtration pressure, or NFP.
Let's call this the fight for filtration.
Yeah.
So what force is pushing fluid out of the blood?
That's the glomerular hydrostatic pressure, or GHP. It's the blood pressure inside that capillary bed, and it's unusually high, around 50 millimeters of mercury.
And why is it so high?
Because the exit pipe, the efferent arteriole, is intentionally smaller than the entrance pipe, the afferent arteriole. It's like blocking the drain in a sink. You back up the water and increase the pressure inside.
Okay, so GHP pushes out. What forces are pushing back, working against it?
Two forces. First, glomerular colloid osmotic pressure, GCOP, which is around 30 millimeter Hg. Remember all those proteins we kept in the blood?
Right, the ones the filter repelled.
They are now pulling water back into the blood vessel through osmosis. And second, there's capsular hydrostatic pressure, CHP, about 10 millimeter Hg. That's just the pressure of the fluid that's already in the capsule, resisting more fluid coming in.
So we have 50 pushing out and 40, that's 30 plus 10 resisting. That leaves a net filtration pressure of only about 10 millimeter Hg.
10 millimeters of mercury. It's a tiny pressure difference driving this massive operation. And it works because those capillaries are inherently leaky, and more importantly, the glomeruli provide a huge surface area, about 6 square meters.
6 square meters, that's like a small rug.
Exactly. It's all about surface area and minimal driving force. Now, maintaining that NFP and GFR is absolutely crucial. If your GFR is too high, you'd pee out your entire plasma volume in minutes, losing water and electrolytes.
That's too low.
Metabolic wastes would accumulate to toxic levels.
So how does the kidney keep GFR constant even when our systemic blood pressure is all over the place?
I stand up quickly, I start jogging.
Primarily through internal controls known as autoregulation. The first mechanism is the myogenic mechanism. The afferent arteriole, that entrance pipe, has muscle around it. If your systemic blood pressure suddenly rises, that muscle stretches.
And it reacts to being stretched.
It automatically triggers the muscle to contract or vasoconstrict.
Like a rubber band snapping back.
Exactly. The pipe shrinks, restricting blood flow into the glomerulus. This automatically drops the GHP, preventing the GFR from spiking. It's an instant pressure dampener.
That's brilliant. What's the second, more complex local mechanism?
That's tubuloglomerular feedback, involving the juxtaglomerular apparatus, or JGA. Think of this as a salt and flow sensor. A sensor for what?
If your GFR increases, the filtrate rushes through the tubules too quickly. Specialized cells called the macula densa sense this rapid flow because they see an increased concentration of sodium and chloride ions.
So the speed and the salt level are the alarm bells.
They are. The macula densa releases chemicals that signal the afferent arteriole to constrict, just like in the myogenic response, but it also signals the efferent arteriole, the exit pipe, to dilate.
Oh, so it works both ends.
Both actions work in concert to decrease the pressure inside the glomerulus, bringing GFR back down.
That's the internal genius.
Yeah.
But sometimes the whole body needs to get involved.
Yeah.
That brings us to hormonal controls, like the RAAS system, the renin-angiotensin-aldosterone system.
The RAA system is a full-scale hormonal mobilization. It's usually triggered when blood pressure drops too low, threatening kidney function. Juxtaglomerular cells sense this drop and release the enzyme renin.
And renin sets off a cascade.
A cascade that ends with the formation of the incredibly powerful hormone, angiotensin II, A3.
Angiotensin II is the crisis manager here. What's its plan?
It has 4 primary targets, all designed to stabilize pressure. First, it's a potent vasoconstrictor of nearly all systemic blood vessels, raising overall blood pressure immediately.
And second.
Second, and this is key, it performs kidney-specific triage. It preferentially constricts the efferent arteriole, the exit pipe.
So it's tightening the drain, backing up blood flow only in the glomerulus.
Precisely. This keeps GHP high, ensuring the filter keeps working and clearing waste, even when overall blood flow is low. Without this, your GFR would just crash.
It sacrifices peripheral blood flow to protect the critical filtering operation. Very clever.
Exactly. And its final 2 actions deal with volume. ATIase stimulates sodium and water reabsorption directly, and it stimulates the release of aldosterone, a hormone that further increases reabsorption of salt and water.
And since this system is so effective at raising pressure, that's why hypertension drugs like ACE inhibitors target it. They're trying to turn down the body's emergency setting.
Correct. But homeostasis isn't just about raising pressure, it's also about lowering it. When your blood volume gets too high, the heart releases its own hormone, atrial natriuretic peptide, or ANP.
The counterbalancing force. How does ANP do its job?
It does the opposite of AC in the glomerulus. It dilates the afferent arteriole, widens the entrance, and constricts the efferent arteriole, tightening the exit. This combination maximizes the pressure difference, which raises GFR and leads to increased fluid loss.
So ANP is basically telling the kidney, we're overloaded, start shedding water now.
That's exactly what it's doing.
Here's where we move from the mechanical genius to the deep history. Why did this system evolve to be so complex? This constant battle between pressure-raising and pressure-lowering hormones. The physiologist Dr. Homer Smith traced this entire journey from fish to philosopher.
And that journey shows that the system we have today is an accumulation of adaptations, often built backwards. Early systems in simple life, like amoebas, were just simple vacuoles. The complex filtration system is actually a relatively recent invention, evolutionarily speaking.
So what was the big leap for vertebrates?
It happened when our vertebrate ancestors, the ostracoderms, moved from the salty hypertonic sea into hypotonic freshwater. Suddenly, they had a huge problem.
Which what?
Water was constantly rushing into their bodies, threatening to swell them up, and their vital salts were leaching out. They were facing constant internal flooding.
So they needed a way to bail water out fast.
Precisely. And that pressure to rapidly excrete excess water and conserve salt drove the evolution of the high-pressure glomerulus. It was essentially bolted onto the existing tubular system to achieve that massive, rapid filtration rate we talked about.
It solved the flooding problem. But then, life got complicated again when some species moved back to the sea or onto dry land.
Right, where water conservation was suddenly the priority. The fish that returned to the ocean faced dehydration. Filtering 180 liters a day was now a huge liability.
So what did they do?
Many bony fish, like the seahorse, slowly lost their glomeruli entirely. They became aglomerular. They just switched back to relying on simple secretion and reabsorption.
And sharks found a truly bizarre workaround.
Bizarre, but ingenious. Instead of filtering, they retained huge amounts of urea waste product for us in their blood. This made their blood osmolarity slightly higher than the seawater around them.
So they could absorb pure water through their gills passively.
Instead of fighting the ocean's osmotic gradient, it's an incredible adaptation.
Talk about recycling your waste. Now, for terrestrial life, where water is always scarce, reptiles and birds made a similar waste adaptation.
They shifted their protein metabolism to produce uric acid, which is excreted as a paste. It uses minimal water. But they still lacked the key mammalian innovation, the loop of Henle.
Which brings us to the mammalian secret, the ability to form urine that's far more concentrated than blood, the ultimate water conservation tool.
And that ability is entirely thanks to the unique geometry of the loop of Henle. We call this entire setup a highly efficient active osmotic exchanger.
So explain the countercurrent mechanism that makes this work.
Well, think of it as a U-shaped tube. The descending side of the U is highly permeable to water. As fluid goes down into the salty, deep medulla of the kidney, water is just pulled out by this concentration gradient.
And the ascending side.
The ascending side of the U is impermeable to water. Instead, it actively pumps massive amounts of salt out into that surrounding environment, constantly reinforcing the salt gradient that pulled the water out in the first place.
It's a self-sustaining cycle. It creates the very salty environment that it needs to operate.
Exactly. And this countercurrent principle is incredibly energy efficient. The kidney separates waste from 200 liters of water at a staggeringly low energy cost. Standard industrial filtration, like reverse osmosis, requires 200 times more energy.
So why is the kidney so much better than our own technology?
The secret is its double loop geometry, the Henle loop coupled with the collecting duct. If the kidney only used that single U-loop, mathematical models show we would still lose about 18 liters of water every single day.
Which is completely unsustainable.
Totally. But by routing the final fluid through the collecting duct, a second water-permeable loop that harvests the same osmotic gradient, the system's efficiency is essentially multiplied. It's squared.
Squared efficiency.
Huh.
That allows us to reduce our daily water loss down to the 2 liters, or so we actually see. That's the genius of using the same energy source twice. This system is a marvel, but it's not infallible. We see a lot of common urinary system challenges, and they often trace back to a failure in one of these functions.
Oh, absolutely. Take infections. Urinary tract infections, or UTIs, are usually bacterial. They lead to painful, frequent urination. The most common is cystitis, a bladder infection, when bacteria ascend the urethra.
And then there are mechanical failures. the dreaded kidney stone.
Nephrolithiasis, yes. These are hard deposits of mineral and salt, usually calcium oxalate, that form when the filtrate is supersaturated. If they get stuck, they cause renal colic pain, which is famously among the worst pain a person can experience.
A failure of the crystallization balance.
Right, usually due to low water intake or certain dietary factors.
And what about the long-term systemic failure of the whole operation?
That's chronic kidney disease, or CKD. This is a degenerative, irreversible loss of filtering ability. It's often silently driven by things like high blood pressure and diabetes mellitus.
And those conditions damage the small blood vessels of the glomeruli.
Relentlessly. When GFR drops too low, waste products build up, causing uremia and severe imbalances that eventually require dialysis. It's a complete failure of regulatory capacity.
We also see failures in those highly selective filters you described earlier.
That's a key clinical connection. Glomerulonephritis is inflammation of the glomeruli. If those 3 filtering layers become damaged or inflamed, they get leaky. Suddenly, the insurance policy fails, and you start seeing large blood cells and proteins in the urine.
Which should never be there.
Never.
And finally, a classic sign of reabsorption failure. Glycosuria.
The appearance of glucose in the urine. It's a perfect example of an overwhelmed system. Normally, 100% of filtered glucose is reabsorbed, but in uncontrolled diabetes, blood glucose levels are so high that they just overwhelm the reabsorption proteins.
The kidney can't grab the glucose fast enough.
It gets left behind in the filtrate.
So clinicians use tools like urinalysis to check on function. But for a true measure of health, they look to renal clearance.
Yes. Renal clearance measures the rate at which the kidneys remove a substance from the blood. And to get the most accurate GFR, they use a substance called inulin.
Why inulin?
Because it's filtered perfectly by the glomerulus, but is neither reabsorbed nor secreted by the tubule. So its excretion rate equals the true filtering rate of the kidney. It's the gold standard.
So we've traced this system from the microscopic 3-layer filter through the high-stakes hormonal triage of RAS, and finally into the evolutionary pressures that created this low-energy, double-loop osmotic exchanger. It's an elegant system.
It is, and that balance highlights a fundamental conflict that was forged in that ancient move from the sea to freshwater. The high-pressure glomerulus was an adaptation to excrete water. It requires the heart to work overtime, pumping a quarter of its entire output into the kidneys.
But the tubules then evolved to aggressively reabsorb 99% of that filtered water.
Exactly. So the heart and the kidneys are locked in this constant conflict. The heart is driving immense volume in, and the tubules are scrambling to pull almost all of it back out just to prevent catastrophic loss.
A constant struggle.
Precisely. So, given that the Earth's environment is always changing, pushing organisms into new osmotic and pressure situations, and the human kidney is still largely a collection of these historical adaptations, the question remains for you to consider. Is the human kidney system truly perfected, or is it merely the best Rube Goldberg machine evolution could build so far in our constant struggle for homeostasis?